So it's my pleasure to introduce Dr. Matthew Posewitz. He's a research faculty here at Cover School of Mines, Environmental Science and Engineering, and also visiting research scientist at the National Renewable Energy Lab, where he is a postdoc looking at algal biofuels. So we're lucky to have him here today. And, excited.
Thank you very much, John. It's a great pleasure. This is quite a group, and it's very nice to see all the interest that's emerging in the grassroots of this. I noticed that at Yahoo yesterday, they always list the top 10 searches that are on the website today, and biodiesel was number 2 yesterday. So I don't know if that had anything to do with this particular conference or not. It's pretty exciting to see all that interest come out. What I'm involved in working closely with Jonathan, he's one of the graduate students that's associated with the project, is sort of a reemergence of a Department of Energy project that went for about 25 years in the 80s and closed down eventually in the late 90s. And that was their aquatic species program. And that was an effort to look for new feedstocks for renewable bioenergy resources. And this has kind of died out after a funding crisis in the 90s, but it's been reinvigorated. And in biotechnology and in engineering and all these other things, great advances and strides have been made in the last few years. And subsequently, there have been a number of recent initiatives by the Department of Energy and the National Science Foundation to fund a whole variety of projects in the microalgae. And the microalgae are obviously all the vascular feedstocks that people have been talking about in the land crops. And one of the main attractions of these aquatic species is that they have extremely high photon conversion efficiency. So it's the rate in which you convert photons from the sun through photosynthesis into bioenergy. Another advantage of them is you don't need fresh water to grow them. And they can grow in a lot of variety of waste streams or salt water or ocean water. And those resources are deemed to probably be more abundant for this kind of bioenergy feedstock purposes than is freshwater and freshwater resources. So currently within the state of Colorado, there's a new center that has emerged. It's the Colorado Center for Biorefining and Biofuels. And it's a collaboratory between the University of Colorado, Colorado State University, CSM here, and the National Renewable Energy Lab. And amongst those 4 institutions, there's a variety of expertise, both in metabolism of organisms, in photosynthesis, in energy, in bioenergy processes. And what we are trying to do. And so what we're really looking at is a process. Photosynthesis slide doesn't show up as some sort of Macintosh PC issue. But essentially what we really like to look at is the conversion of photons. And that involves isolating very robust strains that are able to grow very quickly in these microalgae, that have very high photosynthetic efficiencies, and then can go into a diversity of carbon storage products. And then we're looking at biofuel conversion, and then we want to refine all the biomass that's present in those fuel molecules. And then we want to eliminate our fresh water, and then go after a diversity of inorganic element recovery techniques. Now, in the late 80s, this was sort of the end-all project down in Roswell, New Mexico, and it was big open paddle wheel type projects. And people do cultivate these algae in open systems, and there are farms that were built. Technology is still evolving, and I think we're going to hear some more about this shortly. And so why would anyone want to use algae in addition to the photosynthesis rates? It turns out that they have a diversity of carbon storage products, and that includes lipids as well as starch. The lipids, obviously, is what a lot of the people here at the Biodiesel conference are the most interested in. But we're looking at it from a variety of other standpoints as well. Interestingly, these algae, in addition to fixing CO2, under certain conditions can be tricked to make hydrogen rather than do CO2 reduction. So they have a lot of metabolic diversity that we really want to probe in and tap into. The other aspect is we can grow these in water areas and waste streams in places where it's not going to compete with current crop foods. And that's going to be important to us as well. We don't want to make sure that we're not going to compete with current agricultural crops, that we can go to new locations and that we can grow these things in the desert or in coastal areas where there's not agriculture at the moment. So if you take a look at different types of algae, You see that under certain conditions they can accumulate about 50% of their dry weight as triglycerides when they're induced by a nutrient stress. And the fatty acid compositions, depending on the strains of algae that you're looking at, which typically go up and down in columns, can vary quite dramatically from species to species. And because we get those high variabilities of the kinds of acids that we can accumulate and the kinds of carbon that is fixed by them, we really want to go in and find out which species of algae are the best for making certain kinds of fuel feedstocks. Now, these algae, in addition to doing photosynthesis, and this is really where we come into this, is at the front end of this, is how can we take photons and oxidize water? And from a chemical standpoint, that's an extraordinary reaction. That's an extraordinarily difficult thing to take water and oxidize it into oxygen and electrons. But once you've mobilized those electrons, they go up into a carbon fixation pathway. And this is where there's great diversity among the algae, whether we go to starch or whether we go to fatty acids or lipids. Now once that carbon's fixed, we can go into two different branches within the algae. Most terrestrial plants do not have this fermentation side available to us. It all goes to this aerobic side, which means you have oxygen present, versus the anaerobic where it's gone. But in this oxidative carbon metabolism, this is where you're accumulating your starch or your lipids. And then these organisms also do a little bit of respiration, as do vascular plants. And respiration is kind of a downside to the whole process, because we certainly don't want to harvest photons for photosynthesis and then go through a respiratory process that gets us essentially right back to where we started. So what we're really going to look for are strains that put our carbon fixation products into good feedstocks, the appropriate carbon chain lengths for lipids, biodiesel fuel, or to starch, or to other commodities that we can use. Now, there's another pathway in these algae, which I'm not going to talk about too much today, but they can go into alcohols and organic acids. So we can actually do some one-stop shopping with these organisms when we go from the photosynthesis, the fixation of CO2, to the fermentation of alcohols and organic acids. Now the current concept is we want to cultivate a bunch of strains that grow in extreme conditions, not using fresh water. And then we want to see who grows the fastest, who's robust. There's a variety of techniques that we use to assess photosynthetic efficiency. Any successful biofuel crop is really going to be about the efficiency with which you can harvest the photon from the sun and convert it into a fixed molecule of CO2. Then we want to characterize the distribution. Any one of these organisms, including as Sam talked about earlier, some of these other plant species, they're going to accumulate three basic types of fixed carbon. One is going to be lipid, we're going to have carbohydrate, which is your starch, and they're also going to have proteins. Now photosynthesis, these organisms have gone through an enormous amount of work to fix all this CO2. And so we don't want to just go after the lipid and then leave the carbohydrate and the protein on the table. We have to know what each one of these species is accumulating and how to go after it from a fuel standpoint. So then once we've characterized the lipids, the carbohydrates, and the proteins, we want to look at the algal production of the extraction techniques that was touched on earlier and get that into the lipids and the biodiesel. We want to take the carbohydrates and the proteins that are left and put those into a fermentative pathway, either endogenously within the same organism or into a consortia of thermophilic and mesophilic organisms that are able to ferment carbohydrates and the proteins into methane, hydrogen, alcohols, and organic acids. And that way we're looking at the whole organism, all the feedstock that's present in it, converting it all to a useful fuel at the end of the day. So what's known about these microalgae? It turns out that there's an enormous amount of information available on them. They've been studied extensively for well over 100 years, and one of the things that we've been in is a hydrogen production assay with some of these algae for quite some time. And the first reported hydrogen production was done in the 1890s in a paper submitted to the Massachusetts Board of Health on the odors and tastes of surface waters. And they noted in one of those species that if they put it in a jar and closed it, it evolved hydrogen gas for several days. So there's huge collections of these species all over the place, and this is a group that we collaborate with in Hawaii. And this is just rack and rack and rack, test tube after test tube after test tube of different species that have been collected from all over the world. We're collaborating with this group. They have cyanobacteria, which is a little bit different than some of the eukaryotic algae. And they have about 1,800 viable strains that were collected from all kinds of places in the world. Oceans, and hot springs, and acid lakes, and basic lakes, and salt lakes. And we're going to go through those in a systematic fashion to figure out which one of those strains is capable of producing. We really want things that grow fast so that we're getting good photon conversion efficiency. And then we want to do an assessment of what kind of fuel molecules we can get out of there. The Hawaii group also houses the NREL collection. That was a collection that was the first iteration of this aquatic species program, which was started so long ago. At one point, this group had collected over 3,000 strains, but because of the budget cuts, they've lost most of them. It's down to about 180 now. So they're kind of reinventing this wheel again, but it's a worthwhile endeavor. They also have the Mitsui collection, which was a professor at the University of Miami that collected a variety of marine cyanobacteria, and that was done in the anticipation of using ocean water to cultivate things. One of the places that we're looking right now for feedstocks for this is the Great Salt Lake. And this is a train track that actually runs across the middle of the lake. There's a train that's going across it. The lake is busted up into two arms. That's the north arm, that's the south arm. They thought they put culverts in this dike here that the water would mix and go back and forth into the homogeneous lake. That didn't happen. When the salt got in there, it crystallized out, and it plugged it up, so there's no exchange. So what you have here is a lake that's 30% salt, saturated salt. On a hot day, it's coming out of solution. And then down below here, you have salinities that range from 3% salt to about 22% salt. The reason we got interested in this kind of work was that if we were going to take ocean water, seawater, and put it in some sort of desert region and make an open pond of these algae, as it sat in the sun, it would dehydrate. And as it dehydrated, the salts would remain behind. So you would need some kind of feedstock that could survive that concentration of salt. And so we went in here to see what we could find and do an inventory of what was available out there. And you can see that dike that runs right across there, and that's the north arm, that's the south arm, and Salt Lake City sits here. It's a very weird place. It's a very strange place when you go there, for sure. And it's one of the sites we went to was actually oil seeping, bubbling out of the ground into the lake, and microbes all over the place making a living off of that. But we isolated a whole bunch of species from all kinds of different salinities, ranging from seawater to saturation. This is the crew that we took out there. There's Jonathan. He did a great job out there. We took our light meter and measured exactly the photons that were hitting the surface and what the photons were at different depths. And then we collected water samples from each different depth to try to figure out what photosynthetic organism was living here, what it was doing with the photons from sunlight, how we could eventually convert that into a fuel molecule. We worked with the Department of DWR out with the Department of Wildlife Resources. We give us access to the boats, which is a pretty big deal because when you put a boat to the Great Salt Lake, corrosion is a major problem. So it's nice to have somebody that's got all this stuff set up. But in our first trip, these are the organisms that we've collected. And you can see just from the morphology of them that there's a very weird and distinct groupings in all these. There's all kinds of diversity in life that exists out there. And here's a little corkscrew guy, Spirulina. And we're interested in that organism because we don't need to centrifuge the cells to collect them. We use a net, a fine net to go through there. That would reduce our cost of harvesting the algae. And there's a number of diatoms. And diatoms are proposed to be a very good feedstock for obtaining lipids out of. And there's all kinds of green algae, which predominantly are good starch producers, but some accumulate high concentrations of lipids as well. But just going through an area like this, which a lot of people associate with not being a very alive or biodiverse ecosystem, it's just full of all kinds of different forms of life. We really want to find out what we can do with this. And then we also have an active group that looks at the whole photosynthetic process. We have a bunch of different techniques that we can look at for light harvesting, photosynthetic efficiencies, and measure which ones are using the best utility out of the photons in the sun. So the basic plan is look at these microalgae or even microalgae, and then we want to look at all the fuels we can get out of them. Hydrogen, lipids for biodiesel, carbohydrates for ethanol, and other kinds of alcohols and organic acids, and then whatever biomass or whatnot is left over, we can even look at chemical processes for the conversion into fuel. This is sort of the prototype green algae that everybody studies. It's called Chlamydomonas rhynohardi. And this is just kind of an entree into how the times have changed for this whole field. There was a long time where you couldn't really do genetics or microengineering in these. This is a mutant that we did some engineering on. We knocked out one gene, and this is what Chlamydomonas looks like when it's done growing. It's just a big pack of starch, and that's really what we're after to go after for the biofuels. With one mutant, one gene deleted, we made that mutant not accumulate any starch. It's wiped it out completely. And so we're interested in this because if there's actually a branch in these microorganisms between, is it going to accumulate lipids or is it going to accumulate carbohydrates? And in the initial iteration of the Aquatic Species Program, it was a major task to find out which gene controlled whether or not the fixed carbon went to starch or went to lipids. And so we've got those things in hand now. We're learning lots and lots more about the genes that are in the organisms. The other thing that we've learned from these things is that if we take away certain nutrients, in this case it's sulfate, that if we take away sulfur, sulfur is essential for making proteins and making cells divide. But if we take that away, they no longer do cell division, they just do CO2 fixation, which is what we're after. And you can see in this strain, as soon as we take the sulfate away, the fixed carbon goes up well over 10-fold. It really starts to pack it on. It's a small movement. So we're getting all kinds of little ways to learn how to grow them, how to culture them. There's other techniques that are available. This is one of our microarray chips. What this allows us to do is the entire genome is plated on a microscope slide that'll fit in your palm. And this organism has 16,000 genes, and you can take one sample, put it on that slide, you get a readout of every single gene in that organism, whether it's on or whether it's off. And as this information becomes available to us, we have a lot more leverage to start to understand these organisms, how to get into these metabolic pathways, go after the sort of fuel stocks and feedstocks that we're after to make bioenergy. So how are we really going to make this work? What can we really expect out of it? Photosynthesis, just based on some fundamental principles. It's a two-photon system. And that means that right away you're only going to be 50% efficient for what's in sunlight. The second disadvantage it has is it only uses about half the solar spectrum. The other half of the energy in the solar spectrum isn't utilized by the planet. And there's a third disadvantage in which the stable charged separated entities are of less energy than the wavelengths of photons used to make that reaction go forward. So the best we can really hope for in photosynthesis is about a 15% efficiency of photons to fuel. But algae have the highest conversion efficiencies of photons to fuel. And the benchmark that we shoot for is about a 10% efficiency threshold. And if we can get 10% efficiency out of any feedstock, but primarily algae is what we're looking at right now, you would need something about 10,000 square miles to satisfy our gasoline requirements, what we use now. So it's a big deal. It's a lot of ground. But when you map it out, it becomes a much more tractable issue. The other thing that you can see from this is if we're smart enough and clever enough to figure out ways to get this cultivation offshore or in very arid and desert regions, this really starts to reduce the demands that we place on our current landscape for bioenergy production. So this is all just very much a theoretical number right now. We can probably operate practically about 1%, so we need about a 10-fold improvement. But it's not out of the realm of possibility. And it's certainly within the scope of the theoretical limits of photosynthesis and what it can do. So one of the places we're really looking is these little guys here. They're very incredible little things. They're diversity of metabolisms, all kinds of different feedstocks in them. But we need to know more about them, how to grow them, how to culture them, how to cultivate them, how to trick them into making the right feedstocks that we want, and then how to develop the right refining and processing techniques to get those bioenergy feedstocks back out. So the initials program was started in 1996, and it all shut down, unfortunately, at one point, but it's all going to be restarted back up. And that's one of the labs, and that's all the algae. Everything's about lights, photons. And I think algae have a real distinct advantage because they're able to take sunlight and CO2, as most plants are, but you can also bubble in these aquatic species a fixed CO2 stream through it for growth. You also get the appropriate feedstock representatives and the lipids for biofuels, and then you have the advantage of being away from freshwater resources. And within this even small field, there's lots and lots of people that are coming in and growing. There's lots of companies that are starting up, lots of universities that are entering this, and a lot of national labs. So there's going to be a pretty high effort, high impact area going into all of this. I think in summary, nature's come very close to solving this whole problem. I mean, it does all the chemistry that we want it to do. It can take light, it can take water, and it can fix CO2. We just need to put things in a different context and figure out how to leverage all of what nature's come up in its innovation for and drive it a little bit more over the top for our energy production needs. All the enzymes and proteins already exist. We just need to put them in novel context or give them some improved properties. The diversity of microalgae is totally unexplored. It's an era of rapid biotechnological techniques that we can leverage for this cause. And there's a whole portfolio of fuel molecules that we can look at to get out of biological systems. Biodiesel, butanol, methane, ethanol, hydrogen are all tractable right now. So there's a whole bunch of people that's particularly involved in our effort, both here at the Colorado School of Mines, at NREL, groups at Princeton and Stanford, Montana State, and Hawaii. And it really comes down to looking at all these different ways, photosynthesis in particular, how we can get those electrons, either the hydrogen or the CO2 fixation pathways, what all these different organisms are that are out there, what are the enzymes, how can we use those enzymes to meet our needs, and then figuring out metabolisms, metabolic pathways, and then doing all kinds of cultivation and other ways to improve our feedstock availability. So I thank you for your attention, and it's a great pleasure to see you. I'll take any questions.